The overall goal of the following experiment is to assess the contributions of colonization and its associated damage to the initiation of innate immune responses in C. elegans. This is achieved by exposing a synchronized population of young adult C. elegans to Pseudomonas aeruginosa, expressing green fluorescent protein, or GFP, which produces a population of worms variably colonized by the pathogen. After washing, the differentially colonized populations of worms are separated into pure populations, focusing on the two extremes of fully colonized and non-colonized worms. Next, RNA is extracted from these populations, and immune gene expression is evaluated in comparison to worms grown on non-pathogenic E. coli in order to examine the relationship between the immune response and colonization. Results are obtained to show that the innate immune response of C. elegans to P. aeruginosa can be decoupled from pathogen colonization.
The main advantage of this technique over other approaches for studying initiation of immune responses in C. elegans is that it addresses the variability in infection in worm populations, allowing us to tease apart different aspects of the infection process.
This method can help answer key questions in C. elegans innate immunity, such as what is the basis for initiation of innate immune responses, and in a high-throughput manner, what's the impact of different genetic or environmental manipulations on the ability to resist pathogen colonization.
To begin, grow worms on several nematode growth medium or NGM plates seeded with E. coli bacteria.